goes in a small thermal cycler and includes a repeat of procedural 3 steps. Denaturation. The DNA sample is heated to separate it into 2 strands, and annealing. DNA primers attached to the opposite ends of the target sequence. Elongation. Heat-tolerant DNA prime. DNA is to copy the 2 strands. This is the diagram of the polymerase chain reaction.
Okay, so basically, as you said, in the beginning there's a process called denaturation. What we do in basically we get the DNA. There's a machine that looks more or less like this. A big machine with a big machine with a lot of buttons, and there's like a thing over here where we put the DNA in the samples, and we heat it up at 9 degrees from a temperature that goes from 78 degrees to 9 degrees Celsius. After that, the nitrogen bases get denaturized, and the hydrogen bonds between the base pairs they break. And there's also a polymerase that the DNA polymerase that breaks the bonds between the DNA. After that, there's like over here another place where you put the new nitrogen base pairs that will go and do and form bond to the new to the old strands of DNA, so to form new ones. And so you introduce nitrogen base pairs, you have the new DNA strand, and it keeps on going and going and going until from like this tiny bit of DNA you have much, much more. So basically, let's say in a crime scene, we have a little piece of paper, we need more DNA. We get that DNA, we divide it, we heat it up, we put more nitrogen base pairs in, and all we do at the end, we have like 10 times or 100 times more the DNA we originally started with. So that's really good, and it also tells us, it is also used with electrophoresis, which we're going to speak about later, to also determine whether or not like a child is really, it was really born from like determined parents. So we're going to speak about electrophoresis. You can get the definition.
So the definition of electrophoresis. It's a technique for separating protein molecules of varying sizes in a mixture by moving them through a block of gel. And that block of gel is usually agarose. By means of an electric field with smaller molecules moving faster and therefore farther than larger ones. So why is it gel electrophoresis used? It is mostly used to provide information that is genetic, and it's also used to analyze DNA, provide evidence for law enforcement, and diagnose diseases that are genetic, and provide paternity information.
Okay, so this is our electrophoresis machine, if you can see that. So this is, the red one should be the cathode, I guess, and the black one is the anode. So this one's negative and this one's positive. As we should know, the DNA, because of the nitrogen base pairs, has a negative conductivity. Therefore, it will tend to go towards the positive if there's a battery between these. So on the blackboard, it looks like this. So we have the machine, and there's these little triangles that they're called wells. Sorry, rectangles. So what we do, we get like a pipette and we put the DNA on the wells, and we have a cathode, and an anode over here, and there's like a battery that connects it. So once this happens, there's like a gel that we use. We can either use the one gel that's called agarose.
really similar to gelatin, and its consistency is really resistant. So it's good for the machine because it helps the pieces of DNA as they try to move through the gel. So it keeps them really compact to each other.
Or otherwise, if we're trying to divide proteins which are like smaller, we can use SDS, that stands for sodium dodecyl sulfate. But we usually use that if we want to divide proteins that are like, or DNA strands that are like 50 or less, they have 50 or less DNA, nitrogen base pairs in. So normally we will use agavos for DNA strands that have like 400, 100. So what happens, we put the DNA here, and if we're doing like a paternity test, the mother's one here and the father's one here, we start it up, and the DNA from here, after a while, like after half an hour or an hour, gets divided and splits into groups, like into lines. So like the father, one here, there's one here, one here, there's one here, one here, one here. So what happens? The DNA moves towards the anode, which is positive, and each strand represents how long the DNA strand is. So we use like a DNA ladder, it's called, that has like different, it tells you exactly the levels that how many like nitrogen base pairs are there in, because the bigger the DNA strands are, the harder they are to move from the negative side to the positive side. And then let's say this is the children one. Like we have, I don't know, this one should be, I don't know, we're using agavos. This one has 400, then 200, then 150, and then 100. And this one's the father's one and the mother's one. In this case, we know that the child is actually, the parents are, they're the right ones, because if we compare like where the DNA has split up, we have the same lines at the end, right? But let's say there was perhaps a line over here. So in this case, no parents have a line DNA strand that is over here, and the child has. So neither the mother or the father has it. Therefore, the child must have obtained a DNA from another, either mother or father, right? Or both. And so this is why electrophoresis is used, and basically it just divides the long base pairs of DNA. So here's an example that we can see. So as we can see, like after 10 minutes, it divides a bit. Imagine this, like we have the DNA ladder over here. 20 minutes, splits more. 30 minutes, spreads even more. Do you have another slide? Okay. So that's all.
Before they step down, pull out your devices, anything you're taking notes on that are not on paper. I see some of you writing notes on paper, that's great. Pull out devices. Any questions you have for them right now? Okay, this is a good thing. If you have questions and you want to take notes, you can ask them questions and they can repeat back. Are there questions that anyone has for them?
Yeah, sure. So, okay, DNA is negative because of the nitrogen base.
So we're going to talk about DNA profiling. It's by Minnie, Lucy, Abby, and Danny.
Our presentation is also going to include DNA profiling, a brief DNA profiling, and gel electrophoresis. Yeah, that the first group presented. So what is DNA profiling? DNA profiling is also known as DNA fingerprinting. It is a technique that compares DNA from different sources without mapping the entire genetic material of an organism. So if you think about it, it's basically just a profile of your DNA without having all of your genetic makeup listed, and it looks kind of like this. It's most of the time seen on a transparent sheet, and you can see the profiles the DNA patterns of the So, when a small amount of DNA is found on a crime scene, for example, the polymerase chain reaction, also known as PCR, can be used to increase the amount of DNA. PCR is a biochemical technology used to amplify small amounts of DNA, generating thousands to millions of copies of a particular DNA sequence. And so, if a tiny amount of blood is found on a crime scene, then the DNA inside is very limited. And so in order for further investigations to proceed, we will need to amplify the amount of DNA so that we could allow further investigation.
Restriction enzyme is a particular type of enzyme that is used in PCR amplification. So after the hydrogen bonds are broken and the two strands of DNA are separated, the restriction enzyme, also known as endonuclease, cuts DNA into sections, and it recognizes and cuts DNA only at a particular sequence of the organic basis, so that the scientists can study only a section of the DNA instead of the whole thing.
Gel electrophoresis is a technique that is a machine that vibrates and separates the DNA according to size and charge. That separates the section of DNA according to charge and size. This creates a pattern of stripes and bands, just like the photo. And as DNA is a unique thing for everyone, it is highly unlikely to have the same DNA pattern if the two people are unrelated.
So, when the paternity of a child is investigated, the bands found in the child's DNA are compared to those of the mother and alleged father. Since all of the child's DNA comes from the parents, all bands should be found in either parent, either the mother or alleged father, or the father. Or, of course, a parent only gives half of the DNA to its child, so not all of the bands of the child would be found in the child. And if you could, so in figure 429A, you can see that some of the DNA bands are not cannot be attributed to either the father or the mother. So in this case, the alleged father would not be the actual father of the child. However, on the other hand, in 429B, both of the child's bands are attributed to either parent. So in this case, this father, the alleged father, would be very likely to be the father of the child.
Forensic investigations, there are few suspects and one of them has the DNA that matches with the evidence that is found in the crime scene. For example, blood or fingerprints. And the DNA is breached down into the sections by gel electrophoresis. And through the comparing the bands, the identity of the unknown DNA can be revealed. For example, there was this last emperor of Russian family that people believe that they died due to the gunshots during the Russian Revolution. But then some people believe that the remains were not theirs. So the DNA fingerprints brought in, and it was proven that the remains were the Russian family's belongings. And as you can see from this diagram, the sperm DNA matches the DNA bands of suspect 1, but not suspect 2.
Can we go back real quick? I didn't see that. Can you point out what you mean by that?
This is the sperm DNA, these two bands here, and as you can see, it matches exactly with the ones here which belongs to suspect 1. So this indicates that the sperm found in the crime scene is the suspect 1's. And this is another example of the paternity. So the child's DNA bands, it is same as either the mother's or the father's. So this one matches with the male one, and this matches exactly with the suspect 2. So this indicates that suspect 2 is the person. Thank you.
Pull out your devices, you're note-taking things. What questions do you have for them? What questions? Pull out your devices, add in.
And its techniques.
So there are like two different types of gene transfer. One is horizontal gene transfer, also known as lateral gene transfer. It is the transfer of gene between organisms in a manner other than traditional reproduction. And then there's vertical gene transfer, which is the transmission of genes from the parental to an offspring via sexual or asexual
And
reproduction.
when IB talks about gene transfer, it usually is talking about horizontal gene transfer, which is, as Sharon said, transfer of genes through other means than reproduction. So there are various types of gene transfer. The first one is transduction, which is the process in which DNA is transferred from one bacterium to another through a virus. And then through gene transfer agents, these gene transfer agents, also called GTAs. These GTAs are bacteriophage-like elements that are created by bacteriums. And they package random segments of DNAs present in host cells and then send them to recipient cells. And then there's bacterial conjugation, which is where one bacteria connects physically with another bacteria through a pili or other bridge-like thing, and they send the host bacteria sends its plasmid to the other bacteria. And the last one is transformation and transfection. This is the main type of gene transfer that IB would be looking for.
So the requirements are desired gene, bacterial cell, plasmid, host cell, 2 enzymes, restriction enzyme, and DNA ligase.
The restriction enzyme might be also called restriction endonuclease in various websites, but the IB calls it restriction enzyme.
So these are the steps for transformation / transfection gene transfer. So the first one is the plasmid is removed from the bacterial cell. The plasmid is cleaved by restriction enzymes producing sticky ends. And the gene fragments from another organism are cleaved by the same restriction enzyme producing complementary sticky ends.
DNA ligase catalyzes the reconnection of the 2 different fragments of DNA. The recombinant plasmid is inserted into the host cell in this drawing.
So that's like a diagram of
So
all the steps.
It's kind of messy, but this is a bacterial cell, and then the plasmid is pulled out from the bacterial cell, and then this plasmid is a part of this plasmid is cut by the use of restriction enzymes, which I will elaborate on later. Then this part just gets removed out like this, and the desired gene from another organism is put in here through the use of DNA ligase. Then this, what do we call this? This recombinant plasmid is inserted into a host cell through a method generally through heat shock. Yeah, a heat shock method is where a cell is put into low temperature for a while, and then the temperature is suddenly changed to high temperature. This allows the cell to have produce large pores in between their cell membrane, so that this plasmid is able to enter the cell. So this, when the plasmid gets cleaved by restriction enzyme, it produces something called sticky ends. And as you see, sticky ends, this is a part of the plasmid. Like, if I were to draw a plasmid in detail, it would be like this. And it's like that. It's a circle like that. And when this is cleaved by restriction enzymes, it is not cut like this, but it is cut like this. So that your plasmid looks like like your end of the plasmid was like this. So it's like that. Okay. And this sticky end, as I said, is done by restriction enzyme, and then it's put together by DNA ligase. If you still remember DNA ligase, it's used in putting the sticky end
It's
with.
like glue, basically.
Yeah, it's just, you can see it as a glue. It acts as a thing that sticks the sticky end of the original plasmid with the desired gene. And then a real life use is synthesis of Acelius. I'll call this BT. Many of you might have this in your presentation as BT corn. I'm going to give an example as cotton. So cotton is really important for people. Lots of people need cotton for their clothes. But there are some insects that decide to feed on cotton plants. So farmers had decided that they had enough of this. So they decided to use various methods, like they use chemicals, but when they were using chemicals, they realized that when they use these pesticides, these insects evolve along with the pesticides, have to develop new pesticides. And not only this, but it pollutes the environment around it. So they thought that this wasn't a good idea. And then they found BT. This BT is a bacterium that kills caterpillars, toxifies caterpillars that eat it. And by taking the genes of this BT and putting it into a plasmid and then shooting it into plants of cottons, they were able to make cottons poisonous to caterpillars. So when caterpillars were to eat this cotton plants, they would get the same effect as eating BT. So by using this method, farmers were able to protect their cotton and greatly increase their cotton production.
So when genes are transferred between species, the amino acid sequence of polypeptides translated from them is unchanged. Unchanges because the genetic code is universal.
This, although it may not seem like much, it takes up one entire benchmark. Just keep it in mind that the amino acid sequence of polypeptide translated does not change. Just
It's like
keep
a
it in
short
mind.
version of it.
And thank you. That's it. Any questions?
Pull out your notes, your devices if you're taking notes on them. What questions do you have?
Can you explain the steps of the gene transfer again?
Gene transfer? Okay.
That one doesn't work very well. All right, devices away. Ears open, go ahead.
So, I'm assuming you know who we are, and you can read, so. We decided that we wanted to show you a 2-minute trailer of the worst movie idea anyone's ever had, ever, that is somehow loosely related to what we're dealing with, if it ever works.
What? Oh.
Oh.
Oh, no. Guys.
Comes on.
Across this great nation, almost everyone has been affected in one way or another by this terrible tomato onslaught. Mrs. Williams, I understand your husband is missing.
Yes,
Do you think he's dead?
Well,
Will you miss him?
Well,
Will you marry again? Laying in a ditch somewhere, left with both his legs calling your name. You won't have to find another man, you know. You're no spring chicken.
Lives are shattered. The nation is in chaos. Death and destruction sweeps the country. Foursquare Productions presents perhaps the funniest film ever made. Attack of the Killer Tomato. Attack of the Killer Tomatoes. You've never seen a film like it. This is the incredible story of the world's ultimate disaster. Vicious, man-eating tomatoes grow to monstrous proportions. Faced with this unprecedented menace, tomatoes. He's getting close, too close for someone. For something. Who could it be? The girl reporter? The ad executive? The press secretary? Or does it go higher? Join Mason Dixon in a race against time as he battles to save the world from the threat of nature's perfect feeding machine, the killer tomato. Attack of the killer tomatoes. You've never seen anything like it.
They actually made sequels to this.
It was a good movie, by the way.
Really?
No. I don't suggest anyone else watch it. It was good in the 80s.
This is what the IB wants you to know for this. Basically, you have to be able to state 2 examples of uses of genetically modified crops or animals, and you have to discuss pros and cons for 1 example.
GMO is the abbreviation of genetically modified organisms, and these are organisms whose genetic material has been changed using genetic engineering techniques. And this is also known as transgenic organisms, and the modification of GMOs involves mutation, insertion, or deletion of genes. And GMO is produced by several ways. With insertion, the genes come from other species, which is a form of horizontal gene transfer, which Andy's group mentioned. And to do this, the DNA has to be attached to a virus, or physically putting the DNA inside the nucleus. There are other ways, but insertion is the most common way.
Example of genetically modified organisms are genetically engineered crops. They usually have traits that make them more resistant to pests, herbicides, and they sometimes have increased nutritional value. There are products that existed and been withdrawn, and those include the Flavor Saver tomato that had an extended shelf life. The Flavor Saver tomato was the first genetically modified tomato or product with the license for human consumption, and it was sold in the U.S. in 1994, but they stopped producing it in 1997. It was engineered to be more resistant to rotting, and they did that by adding an antisense gene, which interferes with the production of the enzyme polygalacturonase, and that enzyme is involved in the process of food softening. Another species of tomato was made more tolerant to salt in soil by adding the gene for salt tolerance from the Arabidopsis thaliana, and that made it for the tomato plant easier to grow in certain regions where there was high salinity in the soil.
Genetic engineering animals the process is actually really slow and expensive, but there are new technologies that are being made to make this genetic modification easier and more precise. There are 6 different groups that they can be classified into based off of what the intended purpose is. So they are research human uses, to produce industrial or consumer products, to produce products intended for human therapeutic use, to enrich or enhance the animal's interactions with humans, to enhance production of food and food quality traits, and to improve animal health.
So there's one specific example. She mentioned therapeutic uses. So in this case, people with hemophilia, which we've talked about before, have blood that doesn't clot right because they have a lack of a product called, what Sarah has informed me based on her knowledge of Roman numerals, is factor 9. But I called it IX before, but I guess I'll call it 9 now. You can supply them with the protein that they need by basically making transgenic sheep. So you take your sheep and you insert your gene coding for factor 9 into the sheep. And we're assuming this is a female sheep because for male sheep this won't work because male sheep have an inability to produce milk as far as I think I know. If anyone can find a male sheep that can produce milk, please let me know. And you basically add in that gene and it will become associated with the genetic information for milk production, and then you produce milk that will provide people with hemophilia factor 9 so that they can actually clot their blood and that they won't die anytime soon when they get a paper cut.
The pros of GMOs are increased food supply, better food quality and taste. Some GMOs have added vitamins. We know so far that it's mostly safe for human consumption. There are no need for pesticides since they're made to be resistant to them, so this is better for the environment. And producing medication by producing rare proteins in GMOs could be less costly in the long run, and then it produces less pollution than making proteins in labs. And farmers gain more control over their crops and livestock, so there's no need for random breeding. The cons of GMOs are potentially there are new human allergic reactions. They make antibiotic medications less effective. Is it safe for animal consumption? Because there are cases where animals have eaten it and they have died because of it. It may be unsafe for human consumption. The genes may enter the environment and cause changes to the natural ecosystem. A large amount of food supply could become the property of a few big corporations. And if it's safe for animal consumption, insects that are not parasites of the GMOs die from it, like bees land on it and then they die.
So the example the textbook uses is BT corn, and you mentioned BT cotton earlier. It has a gene from Bacillus thuringiensis. I don't remember how it's actually supposed to go, so it's BT from now on. It's basically a naturally occurring bacteria that acts like a pesticide. It can be found in soil and also in insects when they eat things, and it produces a protein that's toxic to insects. And especially the European corn borer, which does not just exist in Europe, so it has applications outside of the geographical area. Basically, in unmodified corn, the borer will take the vascular system of the plant, which is basically the tubes that transport water and nutrients up to the actual plant itself, and they will break them apart. They'll break apart the leaves as well, and then in the end, they'll actually eat the corn. So, bad system overall. So, the pros of having BT corn actually exist is that the damage caused by insects is really reduced because the plant itself is toxic, and that's great because it'll kill everything that would harm it. And BT corn is a little more expensive because of the work that you have to do to put the gene in, but it needs less insecticide, so it saves money in that one way. Non-BT corn you need to check really often because you're never sure exactly if there are going to be pests in there, but BT corn you don't have to check as often, so it's much more efficient. And less insecticide means less negative environmental issues and lower health risks. BT also seems to reduce fungus infections, where the fungus releases mycotoxins, which are poisons, and these poisons can be hazardous to humans and animals when they are eaten, and they can't really be removed by cooking or freezing things. So they're pretty much difficult to get rid of. And the cons of having BT corn, it could kill other insects like bees that are actually necessary for other types of crops to be grown in a sense. And it could have harmful effects on humans because when you think about it, when you use insecticide, you're spraying just the outside of the plant. You can wash it off later and eat it. This is something that's inherently poison. Its very nature has become poison. So you may not be able to eat it without feeling some side effects. Insects could also develop a resistance over time through prolonged exposure and also survival of the fittest. I hope you all know what survival of the fittest is. And
you do? We're going to have to teach it then, good.
over time will also make BT spray, which is used as an insecticide for non-GMO plants, pretty useless. So that's a bummer. And it's difficult to prevent pollen with BT from actually spreading to other fields where there are different relatives, like wild relatives or non-GMO relatives. And this would make the non-GMO corn become GMO corn, and they can no longer sell it as organic, and people who only eat organic food won't be able to buy it. And it would also give the wild corn a real unfair advantage in its own environment. Basically, the wild corn is now basically super corn. No insects will eat it. Anyone who does will die. It'll grow and expand all over its general area. There'll be less habitat for animals, and then over time, this wild corn will just completely ruin everything. And over time, of course, this would decrease biodiversity, which is just the general amount of diversity we have in ecosystems. That's all.
If you want to take any real quick notes or jot down questions you have, we don't have time now, we got to get to lunch. Jot down any questions you have. They will be happy to answer questions right when we get back. I will send you an email. Okay? There's one thing I'd like you to try to do over the weekend, just as some practice for some IB type questions of things that we've gone over. I'll give you some sample paternity tests. You tell me who the father is. I'll give you some gel electrophoresis stuff. You'll tell me fragment lengths. Easy stuff from what we went over. Thank you. Presentation's great. If you need to take the dihybrid cross and you want to take it today, I'm gonna give it to Rachel right now. So for those of you that want to take it, you may at this time. Whatever's good for you. Okay.
For this is to define what cloning is, obviously, and also like finding different ways of cloning, and there's also ethical reasons why or why not we should do cloning or not do cloning. Yeah.
Okay, so first of all is a definition courtesy of BioNinja. It says that cloning is a process by which genetically identical organisms or a group of cells are derived from a single parent cell. So this, I think you would all know, the next are the processes by which cloning can occur. Process number one.
So this one is a somatic cell nuclear transfer. So first, the female animal is treated with hormones to stimulate the egg cell growth. Then from that, well, I have a picture, but I'll just draw it out for you guys. So here, there'll be a somatic cell with the nucleus, and then here is the egg cell with the nucleus as well. And from the somatic cell, we are going to take out the nucleus, and since the somatic cell contains the, what was it, Alvin, the 46 chromosomes, it's enough to have, to contain enough to create this cloning. And for the cell, or the egg cell, it only contains 23, which is it's not enough to contain, enough to create the cloning. So for this, the nucleus will be taken out. So now you will only have the egg cell, and then from the nucleus, you transfer it into the egg cell, and then from the egg cell with the somatic cell nucleus, it will form together, and by using an electric shock, and then from the electric shock, it can be transmitted to the mother to give, so the mother will be able to give birth to the cloning animal or organism, or it can be used for testing. So, transgenics or genetical modification, it's similar to what Santali said, but so genetically modified DNA is injected into an egg cell, and it's shocked with an electric stimulus, which then forces the cell to divide, and then it's placed into a surrogate mother so that it can develop further. And so instead of taking it from a somatic cell, you just modify like DNA the way you want it, and you put it into an egg, and it's then produced in a surrogate mother.
Okay, so in the processes that both Santali and Elizabeth outlined, there is the appearance of the current egg cells. But why do we use egg cells for cloning? The main reason is because egg cells themselves, relative to the size of other cells in your body, are quite large, and so this allows for more energy to be used up in the development of an embryo, where you could put the cloned cells into a surrogate mother. And so, for example, in chickens, the entire chicken develops in a single fertilized egg cell. So here you also have the functions of cloning. Here you can see we've divided them up into three major categories. First of all, you have animal cloning, when you actually take that in and place it in the womb of a surrogate mother so it can birth another animal. And so the functions of that, you can save endangered animals, you can also copy elite animals to preserve the few minority. And then you have embryonic stem cells. This is mainly just for research. And so from there, you don't put it into a surrogate mother to birth another organism. Rather, you just store it in its like divided form for research and also for other medical uses. And so you can make transplantable cells, and you can also replace damaged tissue. In the process that Elizabeth outlined, that has to do with GMOs, a topic which I think Deanna's group outlined. So genetically modified organisms. You have the occurrence of this term, the xenotransplantation, and so that's basically transplanting tissue between members of different species as to create like hybrids in a sense. And then you also have research models from cloning.
So here is an outline of the two different ways of cloning that me and Liz have talked about. Here's the somatic one that I talked about, and then this part is the one that Liz talked about. Okay, so Dolly the sheep was the first mammal to be cloned from an adult somatic cell. She was cloned in Edinburgh, Scotland on July 5th, 1996. So it was made by nuclear transfer, so what Santali talks about. So this sheep had three mothers, so one provided the egg, and then another one the DNA, and another one was the surrogate mother. So yeah, just an interesting fact.
Okay, so this presentation has been pretty boring up to this point, and so we'll show you some fascinating examples of xenotransplantation. Oh my God, what is this? Is this an orange or a frog? Is this an orange or a frog? That's the begging question. Just joking, it's not true, but the next thing we show you is, what is that? That a smiling cow. Quite frightening. Even more frightening than a, killer tomato. And so finally, for our last benchmark, we have the ethics of cloning. Should it be permissible or should it not? So for the argument for, you can have these 3 points. It can be used to cure diseases. It's the way that technology is advancing in the future, and if you're lonely, you can have a friend who has you. And the argument against cloning involves these 3 points. Basically, it's the creation and destruction of embryos, and whether that's ethical or not. You have different technologies that perform the same function, and therefore would not necessarily need. So basically, cloning technology would not necessarily be needed. So things like nuclear reprogramming can in a sense take over the function of cloning, and then there are also malfunctions which of course lead to cancer, as the programming of cells cannot stop it, so that you have, when malfunctions occur and you cannot stop the programming, that's basically cancer.
Thank you.
Any questions for the group?